So we have Emily Arturo presenting today. She's actually out in California. So this is a special guest because normally she would not come drive all the way to Vermont. And she is going to talk about coronavirus is not an invisible enemy. And she's from the La Jolla Institute for Immunology. And she will explain what all of that means. So Emily, I'm going to pass it over to you.
All right. Can you hear me all right? All right. Yes, my name is Emily Arturo, but I am legally Emilia Arturo. And so I've put my name here at the bottom of my title in case after this presentation, any of you become interested in pursuing professionally research in virology, the study of viruses, or immunology, the study of the immune system. Feel free to look me up on LinkedIn under Emilia Arturo. You can email me at earturo@lji.org. You can look me up on Twitter at Moonlight Arturo. I use Twitter exclusively for science communication and communication with my colleagues in immunology, virology, and microscopy, which is the tool that we use to see viruses and parts of the immune system. Like Clemencia said, I'm a postdoctoral research associate at LJI, this La Jolla Institute for Immunology. So a postdoctoral associate means that I recently graduated with my PhD in biochemistry. So before I came to California from Pennsylvania, I hadn't had any formal training in virology, nothing about the immune system. It was just out of curiosity. I had always read things, and I knew that this woman, Erica Ullman Sapphire, who is currently my boss, was doing some awesome stuff, and I wanted to help. I was hired as a structural biologist, which means I am a biologist, but I study in particular the structures of all the proteins that are important. In my graduate work, it was for metabolic enzymes, so enzymes that digest your protein and digest the food that we eat. And so I use various tools to look at those proteins, and there are many reasons why we want to know the details of those proteins. This presentation is going to be focused on coronavirus and its proteins and the immune proteins that we make or that are available through vaccination or through immunotherapeutics to combat it. So I think in some way or another, I've been drafting this sort of presentation as an essay or a PowerPoint presentation since March, I think it was, when I first heard President Trump call the coronavirus an invisible enemy. And as a microscopist who has seen many images of viruses and I've focused for the past year and a half on images of viruses and the proteins on them, I thought, no, this is not an invisible enemy. We actually can see it. And seeing it is actually, and valuing the fact that we can see it is really important because that means that we have a handle on it. There are ways that we can rationally design to attack the virus and to help the immune system fight it. Right, so it was back in March when I took this picture of my lab notebook. The date in my lab notebook is Sunday, March 22nd. And this was the day before all of San Diego was forced in a lockdown, meaning all non-essential personnel could not go out. And being a virologist, being locked at home was an absolutely weird feeling. I mean, I knew that now I had a sort of job security I never knew that I had, but I couldn't go into lab. So the day before the lockdown, all 28 of us in the lab got there and tried to kill some experiments that were going that we couldn't tend to during the lockdown, or to just quickly gather some data that we can bring home to analyze, because nobody knew at that time back in March how long this would last, how long it would be before we could get together again. So it turned out that before March, about 20% of the lab was working on SARS coronavirus. So this is the virus that causes COVID-19. You'll hear me call it SARS-CoV-2. There were about 20% of us who were working on that virus. I was working on it just a little bit at the time, just keeping up machinery that was being used to study aspects of it I'll get into soon. But we couldn't fit all those many people in the lab, so most of us went home. So this is where I work. It's LJI, La Jolla Institute for Immunology in the southern part of California. It's a beautiful place. This building is just one building big with 24 different research facilities inside of it. And every single one of those 24 researchers with their personnel aims their research at some aspect of immunology, like autoimmune disorders or allergies. We focus on viral infection, transmission, and vaccine design. Shown at bottom right is our lab. Back in January, early January, we had an anniversary party. So we had just arrived at LJI probably 6 months before the pandemic hit. We had moved there from the Scripps, and we moved there. Well, firstly, I'll just explain the life and disease thing. So the motto of LJI is life without disease. Which is to say that our goal as a group of researchers is to address the burden on human life of various diseases. So we can address that burden by coming up with therapies or coming up with preventive measures or just figuring out how viruses work so that we could keep ourselves safe. So we had a lab party and we separated ourselves into the life team and the disease team. And unfortunately that day, the disease team won. But anyhow, that's us. So we are a relatively large lab, and Erica Sapphire is here in the disease team. Can you all see my pointer? Okay, because I'll be using it later on. So there she is, and she runs 2 consortia from LGI. There's one that's called a COVIC. So it's a coronavirus immunotherapeutic consortium. So this is a huge collaboration with labs all over the world, actually, who are all aimed at coming up with immunotherapeutics. I'll go into a little bit about that, but it's basically antibodies, figuring out which antibodies from survivors or from immunized individuals or immunized mice or other animals we can use to combat this viral infection early on. We're funded by the Gates Foundation to uncover which of these immunotherapies would be easiest to scale up in a safe and effective manner to distribute to places in the world where their healthcare systems are not as advanced as other places. And that COVIC, the thing on the top, is modeled after the Viral Hemorrhagic Fever Consortium. So in 2013, it was a year before the largest Ebola outbreak to date. And Erica had just decided to call a bunch of collaborators and even a bunch of competitors in industry and in academia all over the world and said, we're not getting our assays right. We're not all coming up with the same answers. We don't all have the same models. How can we work together to come up with the cure for Ebola virus? And so if you don't know what Ebola virus is, there was a recent outbreak now that is just tailing off. It's a horrific disease. It's a hemorrhagic fever disease where symptoms are shown within a few days of infection and people bleed all over the place. It's easy to avoid them. 80% or so of people die from this disease. This is different to coronavirus, right? Where you can go a week showing absolutely no symptoms, but transmitting the disease. And so it's hard to control, but it's nowhere near as deadly or as gruesome. But still, we need some sort of therapy that can be used early on to prevent the damage that even coronavirus can do, even if it doesn't kill you. But so back in January, it was kind of an invisible enemy in that we didn't really have an image of the virus. What I'm showing you here is an image that was commissioned by the Centers for Disease Control from 2 scientific illustrators. So they got together with a bunch of other scientists at the NIH, at the CDC, and all over the world who had studied other types of coronaviruses to try to figure out what does it kind of look like. Because if we have an image, we can at least focus our emotions on it, and scientists can focus our scientific efforts on it. So that was the very first image, and this is the one probably all of you have seen in some color variation in like every newspaper you go to every day. So the illustrators took a pretty good guess at what some of these proteins are on the surface of the virus. So the gray ball is actually all made of membrane, so like fats that are part of this virus. It's an envelope virus that has proteins sticking out of it. And the 3 proteins that are sticking out are the ones your immune system sees, right? So if your immune system sees it, we want to know what they look like so that we can figure out how do survivors manage to detect this virus and get it out of the way and clear it without significant health effects. So these illustrators guessed at where E might be, which is this, it's a kind of ion channel. You don't hear about it in the news because it's really difficult to study. Then there's M, which is a matrix protein, something that that we have some expertise in. And it's a protein we think that goes, it sticks out a little bit on the outside of the virus like here, but it also goes through the membrane and attaches to other things on the inside of the virus. So the virus inside has RNA, DNA, well, actually it's only got RNA, and then it's got other proteins. And so the M we think holds the virus in this kind of ball shape. And then the S, or the spike protein, is the one that I think many of you have heard about. So this is the one that antibody tests are looking for, that all sorts of diagnostic tests are looking for. So there are tests that test for the viral RNA, which is on the inside. And then there are tests that test for the spike, which is on the outside, so presence of the virus. And spike, this S protein, is also the thing that drives the immune system the most. So all the antibodies we know so far that work against this virus work against this spike protein. So in January, we didn't really know what they looked like, but we had a guess. And then in February, the Rocky Mountain Laboratories, which is a national lab in Montana, they had already begun working with monkeys, trying to test out some some other drugs like remdesivir and hydroxychloroquine in their monkeys that had been infected with SARS-CoV-2 virus. And so here they've taken some of the earliest real images of the virus and they've colored it blue, and they've colored the infected kidney cell of this particular monkey brown. But again, we really can't see very much detail in these pictures, so we needed more. The CDC put out several more images in February and March. This particular image shows the SARS-CoV-2 virions all colored blue, just so you can spot them. And then all around it are mucosal tissues. So mucosal tissues are those things, those cells that are present in your eyes, your nose, your throat, your lungs, and your gut. And so if you've ever had a coronavirus test, you've probably had a swab that was stuck up your nose and into the deep parts of your nose, and it's trying to access these very tissues where the coronavirus is replicating. And then they also showed this particular image. These are, again, mucosal tissues with their little finger projections, and these are viruses that we think are going out of the cell. They're egressing. And so these viruses are these round balls with the dark spots on the inside. And so on the very outside of each one of these balls, if you squint sort of, you can kind of see a corona of those spike proteins and maybe other proteins. But again, it's very low resolution. And I mean, these pictures are inspiring and they're interesting and we want to know how it's getting in and getting out, but we also need more detail. And so this is where our lab comes in. Right, so we want to look at the details of these proteins. Now, these proteins are important before I get into how we see it, because these are the proteins that, our immune system sees, but these are also the proteins that the virus can use to get inside of our cells. So this image is just a picture from a review poster that was made in part by Erica, my boss. And so on the left, it shows how various viruses attach to cells. So SARS-CoV-2 and the SARS-1 is highlighted with the red arrow. And so you can see the green spike proteins all around there. And those green spike proteins have very particular and specific affinity for one kind of receptor that we have on just about every tissue, not just the mucosal tissues. It's called ACE2. And ACE2 has other functions I'm not going to go into, but it also kind of accidentally lets in SARS-CoV-2. And other viruses have other ways of hijacking our own cells' receptors, like I'm showing you vaccinia virus, which is smallpox, cowpox. It gets in with its own receptors. Its outside protein is not called spike, it's called GAG. And then the Ebola virus uses several other receptors and co-receptors to get in. and its spike is called GP. And then we have here flu, and its kind of spike protein is called HA, or hemagglutinin. It also uses a different kind of receptor, as does HIV. And then once those viruses latch on to those receptors, which are, didn't evolve for this purpose, but that's how the cells can engulf the virus, and the virus can go on to follow through with infection. All right, so I'm going to do a little slide on protein because every friend and family member who's not a scientist who's heard me talk about what I do has heard me talk about proteins until someone finally said, all I hear when you talk about proteins is steak. And I'm not talking about the proteins necessarily that you aim to eat or that people eat, a lot of to go on keto diets. This is protein that does functions, right? It functions on a virus, it functions in our cells, and it does lots of things. These are the enzymes in our bodies that help us metabolize everything we eat. So they all have protein machinery, right? So animals all have cells that contain proteins. Every single one of our cells contains proteins. Insects like this mosquito contain proteins, and insects, by the way, are probably some of the most prolific vector carriers of viruses that can infect and harm humans, as are bats. Right, we have bacteria. The ones in this picture happen to be the ones that cause tuberculosis, and they have lots of proteins, and they have all the machinery that's required for making proteins. Viruses, of course, have proteins both on the outside and on the inside. Plants even have lots of proteins. In fact, plants are often used as a protein source in the lab and also in diet. And then, the things that we eat, even grains that are on the food pyramid, potatoes and rice, they also have proteins. And another reason why I wanted to bring up this slide is because there are several organisms that do have proteins, that do make proteins, have all the machinery to make lots of proteins and lots of different proteins. We use these cells to actually make viral proteins. So you can get proteins by treating viruses and extracting like these spikes out of the membrane of that virus. But it turns out it's not only dangerous when you're dealing with viruses you don't know how to control or that are super dangerous like Ebola, but you don't get very much out. And so you end up risking the life of the researcher who has to have long exposure to these. So to avoid that, we use a lot of cultured cells from animals, plants, insects, and bacteria to create these virus proteins. And we want to create the virus proteins because we want to see them. In order to see them, and I'll tell you why we want to see them more directly in the next slides, but in order to see them, we can't just look at them like we would at a whale, right? So in the bottom part of this slide, on the right-hand side, are really big things that you can just see by the light of day. And on the left side are really small things you can only see with certain tools like electron microscopy, which I'm going to tell you about next. Right, so small molecules might be small molecule drugs like remdesivir or hydroxychloroquine that we want to see. We want to see where they bind. And all of these small molecules typically bind a protein. So protein is next smallest on the left-hand side of this scale. And these are like the proteins on the surface of a virus that we're really interested in. Looking at whole viruses, unfortunately, just gives us kind of a low resolution image. There's lots of moving parts on a whole virus, so that you can only see kind of a corona. So technology right now is very rapidly evolving, but still our highest resolution information is if we look at pure protein. So we have to use those cells. I showed you from the previous slide, those cells from animals, bacteria, insects, or plants, to pump out a lot of protein for us to be able to study them in isolation. That's our best bet right now at getting really detailed information. So this is generally how electron microscopy works. We get the purified proteins. So I'm starting on the left-hand side, and we put it on a grid, which is shown here next to the head of a match. It's really, really small. And in that grid are several hundred little squares. They kind of serve like bowls where the protein, which is kind of in like a liquid of some kind, is going to fall into those squares, and we either stain it with heavy metals or we freeze it. And then that tiny little thing goes into this rather large microscope. So the microscope I'm showing on this slide is actually kind of medium sized. I'll show you some of the bigger ones. But this microscope, unlike a light microscope, it focuses electrons at the very top of this microscope, so really high up. It just shoots one electron at a time, very high energy, very small wavelength, so you could see these tiny things, these tiny proteins that are in that little grid. And then there's lots of computers everywhere because these instruments are super complicated. There's vacuums you need to keep. You need to keep the radiation out, keep the humidity from getting in to the microscope. So they're rather complicated. And then each individual image, so you can get like 1,000 images from one of those tiny little squares in that tiny little grid. And one image might look like the one I'm showing you here. So this black and white image shows black, which is the carbon, or gray carbon, and the white things in this particular case are antibodies. So I study a kind of arenavirus related to Lassa, which you may have heard of as well. It also causes a hemorrhagic fever like does Ebola. And these antibodies are showing up as white dots. And then the lower arrows, the 2 arrows down there are meant to depict, there's lots of stuff that goes on from one image or thousands of images until you get to a 3D reconstruction of an antibody, which is shown below. So we're not just interested, how do we see the protein? Okay, so this is a little advertisement for what our lab is very proud of recently. So shown at the middle is Erica. So this is our PI, our principal investigator, and Ruben, who runs the microscopy facility we are now building. And they're wearing masks because unfortunately our big microscope, this thing called a Titan Krios, just shown at left, was being built just as the pandemic hit. So we were allowing engineers to go in one at a time, knowing that the Gates Foundation had just entrusted us with this consortium of antibodies from survivors that were just beginning to be organized. And we needed our microscope so we can see, because we had just moved to LGI 6 months before. So there's Erica and Ruben basically climbing inside of the microscope. So this thing is huge. I think it's like 9 or 10 feet tall. And again, the electrons start at the very top of this thing and they shoot down onto one tiny little grid that's about, maybe a fifth of the size of your nail. And from that information, we can get things like the thing I'm showing you on the right. These are actually spike proteins or GP proteins of a Marburg virus. So I didn't extract it out of a Marburg virus, but I used insect cells to produce a lot of them. So this was just a recent image that I took with my phone just the other day when I was at the microscope because it was beautiful. So you can see the little white spots are all of the spike proteins, which turns out looks very much like what we think exists on the surface of the virus as well. So why are we interested in viral proteins? Why do we want to see so many details? I mean, a lot of science is just curiosity, right? You just really want to know how something works and you just like use whatever tool you can possibly find to do it. But knowing the details and using such a huge, expensive, complicated microscope is important because we want to know what is the immune system really seeing. So those people who survived the coronavirus, who survived COVID-19 and now have volunteered their convalescent plasma, which is a thing I think you may have heard about in the news as well. This is a whole bunch of antibodies that this individual has managed to produce in response to a natural infection. And we want to know how they work. So this is a crash course in immunology, super, super quick, one slide. All right, so on the left, within the first few hours after a coronavirus or any virus or any pathogen really gets into your body that is not you and that is a potential threat, there's a whole family of cells that within hours will mobilize to begin to kill it. So these cells have various ways of detecting the virus, let's say. They latch onto the outside of it, they latch onto things that leak out of the virus, or they latch on to things that escape an infected cell that's been destroyed by the virus. And only after many hours and then many days do macrophages and dendritic cells, things that shoot out to go to basically catch any, catch and destroy any viruses that have escaped the innate immune cells on the left, Over days, they will start to activate B cells and T cells. So B cells are probably the most important to us at the moment because those are the ones that produce antibodies. Those are the ones that we want to train with vaccination to continue to produce neutralizing antibodies. So these are antibodies that will work, right? That will neutralize the virus. So I showed you in the last picture that I was imaging some antibodies. But we also, so antibodies, I think in the last picture I showed you, they look like this. They're globs of 3 things, but most of the times in textbooks, you will see them as these Y-shaped things. So on the lower left, I'm circling the bottom part of the Y of an IgG. This is where it would bind an immune cell. So one of those cells that gets triggered when it senses, when your entire immune system senses something going wrong. And the top part of the IgG is what binds the virus. So that's the thing that latches onto the spike more often than most other proteins in a virus. And then there are other kinds of antibodies that are produced, just so you know. IgGs are the ones that we want to make to administer in immunotherapeutics. They're the ones, IgGs are typically the ones that are detected in a test that you might get on campus. IgAs are interesting. They're the ones that get almost exclusively expressed in the gut. And that's the gut versus the rest of the immune system is a whole other very fascinating, complicated thing. IgE here in the middle is a thing that's often linked with allergic reactions. So your immune system kind of going a little haywire. And IgM is this big thing out here, and that's one of the first antibodies that gets produced. So if any of you have had any antigen tests or antibody tests for coronavirus, you will see an IgG marker, an IgM marker, and then some kind of control. So this is what they're measuring in those kinds of tests. So what I'm showing you here again is another antibody. It's an IgG, the Y-shaped thing, right? At the bottom is where it binds to cells. At the top, it's where it binds to the virus. And on the right is shown a crystal structure. I'm not going into crystallography as another method of looking at it, but it's very similar to electron microscopy in that like the little transparent parts are the actual data. So after we get thousands of images that we can reconstruct a 3-dimensional view of the thing, that's this transparent thing that you see. And on the inside, the drawing in red and blue is the atomic model. So once we get really good data, we can find out where every single atom is in the antibody. And there are whole companies dedicated to engineering antibodies to make this part that sticks to the virus more specific. They engineer the bottom part to trigger immune cells more effectively. So seeing these antibodies was really important for us to make immunotherapeutics more effective and safer. Now in March was the first time anyone had seen a real spike from a SARS coronavirus. And this particular paper came out in early March. And for us microscopists, it was amazing. This paper was a collaboration of labs from all over the world. And from the time that they harvested those cells they had used to create that spike protein to the day that they saw this spike protein here on the left, and that they built that atomic model, which is shown here in the color, it only took them 12 days. I mean, for us in the lab, typically working on our own, even in a large lab with 25 people, it sometimes takes us weeks, months, but hit us with a pandemic and this can come out in 2 weeks. And so that was super impressive. And so we saw some really interesting things from this particular paper. Here's the spikes, the thing that sticks out on the viral membrane. So again, there's no virus here. There is no membrane. We just kind of extracted it out of cells that are producing viral protein. And we saw that it binds the receptor. So here on the right, I'm pointing to the ACE2 receptor that is also bound in these images. So we found out where it binds. We found out the details of how it binds. So we can really begin to rationalize what is going on and how can we disturb this. In July of this year was the first time that we saw a real virus at high resolution. Right? So the last image was just the spike, no virus. This image on the left, at least the panel B, is showing whole virions that were taken from a patient. and that were inactivated and put on one of those grids, put in the microscope, shot with electrons, and viewed at pretty high resolution. In panel C are those spikes that are sticking up. So this is it's quite beautiful to see, but it's also really informative because now we know some other things about the spike. And so at the very top of this slide are just, so the data itself is like the gray kind of hazy part, and then we build a model on the inside that best describes it so that we could find out where all the atoms are. And it shows that on the virus surface, on a real virus, you have not just this spike that we saw before, but you have this kind of a long yellow spike too. So they're labeled pre-fusion and post-fusion. So pre-fusion, this is what your immune system sees before it even hits your ACE2 receptor, before it can get in. Post-fusion is supposed to be the thing that exists after fusion. So it was fascinating to us to see that both of these things were present on the virus at once. And then in panel C here in the middle, it just shows that the spike moves. And yeah, the fact that it moves is kind of cool, maybe quirky, but what is fascinating is that this ability to move may be some way that the virus manages to escape immune recognition, right? So some of these surfaces of the virus are only exposed under certain circumstances. And maybe that interferes with antibody binding. I mean, our bodies make antibodies, many antibodies that are not effective, that they don't neutralize the virus. We want to understand those antibodies just as much as we want to understand the ones that do work, because we want to know how this virus also manages to fool our immune systems. So the updated model, right, is shown at bottom right relative to the CDC one. It looks like the spikes, I mean, they did a lot more computation on this, but the updated models show that the spikes are not evenly distributed like this, right? Nature isn't always this beautiful, symmetric, evenly distributed thing. It's kind of a mess. And you can see that in the pink, they're showing, oh, that sometimes the receptor binding part sticks up and sometimes it's down. Like sometimes it's available to bind your cells and sometimes it's not. So that's given us a lot of insight as well. And then in August this year was the first time that someone had published the spike protein, shown here on the left, bound to fragments of antibodies from survivors. So in the middle part and then the right part, they're labeled FABs, F-A-B-S. That just stands for fragments of antibodies. So a lot of us, we cut up the antibodies that we isolate from a vaccinated animal or from a volunteer who survived, just to limit how much information we're getting out of these microscope images. But these two antibodies do neutralize virus. So they did probably contribute to protecting this individual from severe disease. And we think it did it by binding directly, in the middle case, this orange one, directly to the receptor binding. So it blocks the virus from even seeing your receptors. So it can't see your cells, it can't get in. The one on the right, however, is a little weird, and I don't think it's really clear yet how that one binds or how that one manages to protect. Maybe it keeps the spike protein from like moving in the way that it needs to move to get to the receptor. We don't know, but this is really cool to see. And now, as part of the COVID, we're starting to do some of this work ourselves. All right, so we're interested in these proteins because we want to know what the immune system sees, right? And so sometimes we engineer these proteins, these spikes, just so we can see them. Sometimes we want to engineer these proteins because we want to use them in a vaccine, or we want to understand how a vaccine works. I mean, many vaccines that are operating now, we know that they work, we know that they're safe, but we don't necessarily know all the details of how they work. So this is a super crash course in vaccine design as well. There are about 4 different ways that you can design a vaccine. One is a message, delivering the message. So delivering either DNA that encodes the protein or mRNA that can be used in translation to make the viral proteins. And then your immune system will see the viral proteins and never have to deal with the virus. And below each one of these, I'm going to bring up on the screens, I'll show you in green those vaccines that are in phase 3 trials now. So in vaccine clinical development, there are roughly 3 phases. The third one is the last one. It's the one that has the most individuals enlisted as volunteers. And there are, I believe there are 9 vaccine candidates now in phase 3 trials all over the world. And some of these you probably have heard about, even in today's news, like literally today's news. So Moderna was the first phase. Moderna is a company in the US, and it was it provided the first phase 3 clinical trial for a message type of vaccine. And then a subunit vaccine would involve just delivering the spike itself that has been engineered in a certain way. The Department of Defense currently funds one type of this subunit vaccine. It's colored orange because it's actually in phase 2 trials, so it's not past the phase 2 yet. And then there are vector-based vaccines, and it's colored in green because this particular one is in phase 3 trials, but this is the one that you probably have heard about in the news yesterday and today, where they had to halt the phase 3 trial because one of their volunteers became severely ill. And so anytime there are clinical trials of vaccines, they are halted when individuals become ill so that they can inspect to see whether other people are also getting ill and what could have caused this. So this particular one is a group effort by University of Oxford, AstraZeneca, and some other places. And this one is interesting. You actually deliver a virus. There are many established safe viruses that are of this form, where you deliver an adenovirus. Adenoviruses are one of the ones, like coronaviruses, that cause like typical colds that you get in the winter or the fall. So they're not themselves dangerous, and they can be engineered to express the virus you're really interested in generating protection from. But they do have their other problems. So it'll be interesting to see what will happen today, tomorrow, over the next few weeks with this particular one. And then the last type of vaccine is an inactivated virus. So you can actually take, in this case, SARS-CoV-2 and inactivate it with, I don't know what this current formulation uses. I have here listed a China-based company called Sinovac, which has inactivated the virus and delivered it as a vaccine. So delivering the actual virus, if you can, train the immune system on the whole virus, right? I showed you some of those spikes were kind of moving around. I mean, maybe that's important to properly train the immune system. So the ideal would be to give an inactivated virus that triggers the immune response, gives you the B cells and the immune proteins that you need, the antibodies, but does not give you any of the disease. So that one is also in phase 3 trials as well. So over the next weeks and months, we should find out how all of these are faring. This is from a thing called Virus Tracker, and many organizations have virus trackers if you're interested. I have the link for one of them on my last slide. But yeah, this has never happened in our lifetime, to have so many vaccines generated in such a short time against proteins we know so much about. So it's super exciting times we're living in. Right, so to end this, I just wanted to give you a few things you might like to have on hand. I'll provide these slides as a PDF to the organizers of the class, so you can go here if you like, but Googling is fine. There's so much out there about SARS coronavirus too, and I urge you to stick to the ones that discuss the science. In particular, there is a podcast that I highly recommend. It is an hour, sometimes 2 hours long, but I'm a runner, and so I end up running longer and longer distances just so I can finish this thing. This podcast is over 10 years old, and it has been talking about viruses, for 10 years. And now they have epidemiologists, they have nutritionists, politicians, virologists, immunologists come on this podcast and give really up-to-date information about what's going on in the clinic, in governments, and on the ground, what's going on in laboratories. So I highly recommend it. You can find it anywhere. And then this is a link to the institute where I work, and you can find out more about the COVID if you'd like. Then these two are national laboratories that provided some of the images at the beginning, right? The Rocky Mountain Laboratories is a part of NIAID, which is run by Anthony Fauci. And NIAID is the National Institutes of Allergy and Infectious Diseases. And Rocky Mountain Laboratories is really at the forefront of working at this virus, with this virus and animal models to study it. And then of course there's the CDC. BioRxiv is super interesting. It's usually scientists like to publish their data and analysis only after it's been peer reviewed, after it's been criticized to death, and we're as sure as we possibly can be that we're saying the right things. BioRxiv is not that way. So it actually publishes not peer-reviewed reports, but that can be published really quickly. And in the case of a pandemic, publishing things as quickly as possible without fear of people stealing your ideas is extremely important. And what's even better is that you can get access to it. You don't need any access through your school. You can send this to family members. They can read all of the data. So if you do end up Googling and you run into some stuff that you're questioning, if it's real or not, you can actually find a lot of these reports in BioRxiv now. So you can actually look at the data yourself and the analysis that they've written up and see if it makes sense to you. If you are interested in cryo-electron microscopy or cryo-EM, there's a person named Grant Jensen at Caltech who has put together a course with the funding of the United States government, which is excellent. It's very easy to understand by undergraduates, high schoolers, anyone that's not in the sciences. But if you are in the sciences and you consider a research career and microscopy sounds remotely interesting to you, these presentations are excellent. And then, of course, there's the vaccine tracker that I quoted earlier. The New York Times has a really good one with like a few extra bells and whistles, easier ways of looking at the data. But it'll be interesting to see how we go from 200 vaccine candidates down to, we probably need on the order of 10 or so that can be disseminated worldwide to really give us the right amount of protection. And with that, I'm happy to take any of your questions. I see there's a lot in the chat, but if Clemencia, you want to start off with any in particular, that's fine, and I can start to look as well.
Clemencia, you want me to just start?
Yeah, sure.
Okay, great. Yes, we do have lots of great questions. So the first, let's see, go back to the top here.
I just want to say that if we do run out of time, if you leave me on the Zoom call, I can actually type in answers to these. And then I can either copy them and paste them and send them to you, or you can keep a record of them yourself.
Great, appreciate that, thank you. So our first question goes all the way back to one of your first slides and asking which protein did E represent? If we need to go back to that slide, that was in the very beginning.
Yeah, I'll provide you with the slides as well, but E is that little yellow one. And E stands for, I believe it's envelope, because it's an enveloped virus. It has all this like lipids around it.
Okay, great. Is there any difference in what we're talking about when we say COVID-19 versus SARS-CoV-2 versus coronavirus? And if there isn't really a difference, then why are there so many names?
There is definitely a difference. So SARS-CoV-2 is a virus. SARS initially started off as South Asian respiratory syndrome, coronavirus, and this is the second one. So it's SARS-CoV-2. SARS-CoV-2 is a virus. In the media, I know they call it coronavirus, but there are many, many types of coronavirus, many of which cause nothing but a runny nose. And then there's SARS-CoV-2, which is the thing that causes COVID-19. So COVID is also an acronym, and it's coronavirus infectious or inflammatory disease. I forget now. And the 19 simply stands for 2019 when it first emerged. So they are just referring to two different aspects.
Great. And is SARS-CoV-2 mostly found in the nasal cavity?
So you can actually take samples from people's stool, and it looks like, from what I what I understand, it's difficult to culture the virus from there, but there is sign of virus throughout the mucosal lining. So the eyes, the nose, the throat, the gut, and your intestines.
Most diseases are specific to a species, but some do jump, like COVID-19. What is the difference in protein among those that stay in the same species and those who jump?
So just to correct, COVID-19 is the disease again, and SARS-CoV-2 is the virus that causes COVID-19. We know that SARS-CoV-2 actually can infect cats, dogs, and monkeys. But this particular virus doesn't seem to infect bats, even though the bat SARS coronavirus is most similar in genetic sequence to the SARS-CoV-2. So all I can say with authority to that is there are very finely tuned and evolved parts of the virus that seem to prefer one host over another. But there are viruses that can infect multiple types of mammals all at once, but just because they infect, it doesn't always mean that they cause disease. So we haven't heard of a pandemic among cats, for instance, but there have been cats, like I think early on in the pandemic, there was a tiger, I believe, at the Bronx Zoo that became ill, didn't die, but you could isolate virus that was an isolate similar to that was to the one that's circulating in humans as well. So I guess the answer is it's complicated, but it has something to do with the spike protein and whether it can bind to our ACE2 receptor versus the ACE2 receptors of other creatures.
Next question is about testing methods. One of the students, also Emily, I've had 3 different kinds of tests due to needing to get them every month when I was working in a dentist's office. Are some more accurate, or is it just preference of what the facility giving the test wants?
How interesting that you were working in a dental office, because that's where it's apparently one of the more dangerous places to get exposed because of all the drilling that can generate not just droplets but aerosols that we're starting to think are more dangerous, more easily transmitting the virus than droplets that you get just like by talking. So there are so many tests, I don't even have a list of them. Some of them are FDA approved. Others of them have had emergency authorizations, these EUAs. And they all have different, what's called specificity and different, specificity and kind of their accuracy. They have different ways of talking about it. So specificity means how well can it detect this virus versus all others. And accuracy refers to how well can it pick up the smallest amount of virus. There are tests that test for the viral RNA. Those are typically pretty specific, like on the order of 95%. But then there are tests that test for the spike. So they're looking for active infections. Those are really quick. They don't require waiting days. Those are just like using an antibody, like the ones that I showed you, kind of on a piece of paper. And then you expose that paper to your juices, usually your spit. And if you have virus in there, that would be a positive result. Those are less specific. And they are less accurate, but some of those tests which are less specific or less accurate actually have a place in controlling a pandemic that's as large as this. So we can't afford to wait days to get results. So some of those tests that detect viral RNA are taking too long. We use a technique called PCR. And there aren't enough labs that have enough of this equipment, and there isn't enough time to wait because people are transmitting even when they don't have the symptoms. So some of these quick tests that you could take every day or 2 or 3 times a week, which is actually taking place on several US campuses, would be better. So yeah, there's like tons of companies out there and tons of pros and cons for each one of them. And it is absolutely not clear yet what the best way forward is, except that we need more testing and more rapid testing.
Where did you go to school? And just a clarification that you have your PhD in virology, is that correct?
I do not have a PhD in virology. I have a PhD in biochemistry. So I had looked at proteins involved in metabolic disorders. So this was viewing proteins or engineering proteins for genetic engineering. And so I have a PhD in something unrelated. But for the past year and a half, I've been immersed in this virology lab. I went to school at Drexel University in Philadelphia, and that's where I got my PhD.
We have a couple questions about if you think we're close to a cure or treatment, and if you are specifically working on a cure or a vaccine.
We don't have a vaccine candidate. So our goal was to engineer the SARS-CoV-2 spike protein in a way that was better than the one that was in that March paper. I mean, everyone is trying to engineer it to be more stable, so easy to administer, and that it will survive in your body longer. We are involved in looking for antibodies when we vaccinate mice with the spike protein that we make. So we are involved in that way, but we have no interest right now in developing anything commercial that would be a vaccine that we could scale up. There are lots of people already doing it. So we want to make sure that when there are candidates out there that are good, that we at least have the tools to look at them to figure out how they work. And how close are we from a cure or a treatment? I don't know. I mean, I think it's still legitimately debated how much of the population has been exposed. We did a great job of locking down for a while. And there are many people, it's estimated to be something on the order of 40% who are asymptomatic and may never know that they had it unless they get one of these tests later on looking for signs of an immune response. So unless we know how much of the population has been exposed, we don't know how close we are to herd immunity. If you know what that is, that means if, you know, that's one of the reasons we don't have smallpox anymore. And so many of us already have been, you know, we're immunized against it. And if enough of the population is immunized, you don't get disease. So we don't know that. So I don't know how far away we are from that. And we don't want to infect everyone, you know, to a sufficient level. We prefer to do it via vaccination. And then for a cure, that would mean a drug or some kind of therapy that would prevent the disease. And the immune system, I know I hate to say this, but it is complicated. And it looks, I mean, it's always been this way with every vaccine and every pathogen ever studied, that you can give the same virus to 2 or 5 different people and get different results in them. I mean, our immune systems are responding in ways that have to do with our age, our stage in life, our nutritional status, you know, how much time we spend outside or inside. Super complicated. So I'm pretty sure Anthony Fauci would say similar. It's complicated.
Rachel shared that last year in her cell molecular biology class, she was able to see and look through an electron microscope, and as well as the other microscopes, and that she really liked the experience and thought it was really neat. So
That's great. Yeah. Feel free to get in touch if you ever wanted to, if any of you ever wanted to do research things out here in beautiful La Jolla, California. Let me know.
I think you might have sort of answered this already, but if all the viruses look similar or can have similar traits, why does it take so long to create a new vaccine?
They're not similar enough, basically. So you can't, like the spike of the coronavirus that we're talking about now, SARS-CoV-2, looks absolutely nothing like the hemagglutinin spike of a flu. And so our antibodies do not react to it whatsoever.
Have you found anything else that changed within the virus? If yes, can someone be a carrier without feeling the symptoms and problematic, I'm not sure what it's saying, problematic causes of the virus?
Yeah, there can be asymptomatic individuals, people who, like, a symptom is a thing that you feel. You know, your doctor can't see your symptoms until you tell them about it. So you might have a runny nose and be like, I have no symptoms because I always have allergies or whatever. So it's really hard to tell who has it and who doesn't have it unless they become sick or unless we have tests. So yeah, you can definitely be a carrier without feeling the symptoms. It's the same thing with like herpes and so many other viruses. You can carry it and never know that you have it, never feel ill, and then transmit it. And have I found anything else that changed within the virus? Our laboratory did publish something recently which referred to one particular change in the amino acid sequence. It was one little change, which we're still trying to figure out why it has dominated in viruses here in the US and it's spreading across the world too. It isn't more dangerous. We don't think it makes the virus more dangerous. It doesn't seem to interfere with binding of antibodies we know already work. But it has a change in it. And in some of our assays, we've shown that it causes the virus to replicate a little differently and to present the spike on its surface a little differently. But other than that, we don't know what it means yet.
Have you found any differences in the body's response to the virus and the gut microbiome?
So I wish I knew more about that. I am not an expert in the gut microbiome, but if you do listen to that podcast, This Week in Virology, you will find a lot that's discussed about it. I'm pretty sure that no one has yet confirmed any specific interactions between the microbiome and the immune system's ability to fight off disease. But we do know that each person has a very personalized sort of microbiome. And on top of that, we have a very personalized immune system. So those things together, I would think, predict that we're not going to know about what about certain individuals protects them versus others.
Has any research led us to believe that we are able to get the virus more than once, or do you think that the virus can go dormant and then symptoms flare up again at a later date?
Yes, that is a current hot topic. So there have been 2 publications so far that have detailed reinfection. So there have been stories that clinicians have told, I'm pretty sure my patient tested negative after they were discharged from the hospital, and then they tested positive again. There must be a reinfection. But now there have actually been 2 cases, one of a student, I think I want to say Nevada, but it was a student who definitely did get reinfected because the first time they had COVID-19, the clinicians did take a sample and sequence the virus. And then the second time this person became ill, but less ill, they sequenced the virus that person then harbored, and it was slightly different, meaning that virus probably didn't recombine or reemerge. It was just a different infection. And then there was a similar case in Hong Kong that was described in the same way. But interestingly, in both of those cases, those individuals did not become as ill the second time. So the current hypothesis is that reinfection is possible, but whatever existing immunity via B cells or T cells is able to protect the individual from severe disease.
How long does it usually take to find a cure, or maybe we can add in a vaccine for a virus?
Well, I think this coronavirus will probably show us if we can get it by November 1st, which I still am pretty skeptical about, but November 1st would be 10 months or so, less than 10 months since it arrived in our country. Since the virus was first discovered, we discovered in a patient in our country, and never has that happened. I think for the case of an arenavirus that I study, which is called Hunin or Machupo, to get a vaccine candidate, it took just a candidate, not even phase 3 trials. It took 8 years. And that may be because it was a disease that hit one very small portion of South America, and there wasn't enough of an interest that drove it. Whereas for SARS-CoV-2, everyone in the world is interested. And so it may be going faster than usual just because of interest and dedication of resources. So it's anywhere from 10 months to, yeah, 8 years, 10 years.
When you showed the slide of the types of vaccines currently being tested, how recent are those types of vaccines, and what vaccine type would you see being the most effective?
That virus tracker, I mean, the results that I highlighted as examples down below each type of vaccine, I just got it off the tracker yesterday, and that was updated pretty recently, and I checked on the New York Times one too, and it's the same thing. It's about 9 that are in phase 3 clinical trials somewhere in the world. And as to which I think is going to be the most effective, I think we have to look at it as having multiple vaccines. Not only can no single company scale up enough of the billions of doses that we would need to cover the planet, but we have to do these things in parallel in case one of them goes wrong. Maybe the halt in the AstraZeneca one will be a permanent stop. We don't know. So it's good to have multiple ones going at once, and maybe one will be more effective than the other. I think a lot of what drives which vaccines get to market is funding. So how easy it is, how cheap it is to make something safe and effective. So we'll just have to see.
Great. Have you dealt with any backlash from those who do not believe the science behind the virus? And if so, how have you dealt with this?
I have had no backlash. I mean, I try not to argue on Facebook, for example, with friends or family, but if they do have incorrect information, and I think the person doesn't mind terribly hearing it, I do point out where there are problems in their logic. I did early on in the pandemic call my parents and warn them, no, stay indoors. It's not going to be over by Easter, which is something that the administration was saying. I was very glad to hear that though they were, Republican inclined, they didn't tell me that my ideas were worthless. And so I'm very glad to hear that there are people on all sides of the political spectrum who understand that it is dangerous and that we do have to just be more cautious.
Do you think competition between countries is affecting the production of a vaccine?
Probably. Yeah, I can imagine. I mean, we always have trade wars and physical wars with nations, and I don't see how it doesn't affect our ability to openly share these kinds of resources. So we'll see if we can get along well enough to spread it across the world. I mean, to spread the vaccine, not the virus.
Kira's asking, can you tell us a little bit more about your background, where you're from, and what made you want to pursue studying the coronavirus protein?
Yes, so I'm older than most of my colleagues. Usually by the time someone finishes a PhD, they're kind of like in their late 20s. I'm about 10 years older than that. I got a degree in mathematics actually as an undergraduate. I thought biology was a little boring. I had a really hard time memorizing stuff. Math was easier for me because I was very logical and I didn't have to memorize very much. I just did math. And then I got interested in neural networks, so programming networks, computational networks to detect defects, for example, in helicopter propellers. Those are the kinds of things I was interested in. But at some point, I got really curious about neurons and like real neural networks and how brain cells work and how they communicate. And I realized that they all communicate through proteins, right? All of these channels that line your axons that, allow your muscles to move or allow memories to form. So I got into proteins. And I moved around a lot in the country just because I had a family. So I was just moving wherever the jobs were. But I finally did finish a PhD after 6 years in Philadelphia. And I got interested in X-ray crystallography and microscopy because there is some mathematics in there. It's not mathematics that you have to derive. You just have to be not fearful of it when you see it, getting crunched across your screen. And then I have the power, if I'd like, to interpret the mathematics and understand what the statistics means coming out of all of these thousands of images. But again, you don't have to be a mathematician either. So I interact with people who do have degrees, undergraduate and graduate degrees in biology and in virology, and we're all working on the same thing.
Do you think the virus could mutate and another vaccine will need to be created down the road?
We don't know. So the only mutation we know that has taken form is that one that I mentioned before that has no obvious, gives no obvious advantage to the virus. But we do know that antibodies that survivors generate do neutralize virus with that infection too. So far, it looks like there won't be a problem with vaccines that are developed against the original strain. But sure, it's always possible that another coronavirus that is similar enough to this will emerge and that will be immune, so to speak, itself to our vaccines, but we hope not.
So this is a great follow-up question. Could you use other types of coronaviruses to help create a safe vaccine for SARS-CoV-2 or adapt the vaccines for others to work for this one?
I think so. I don't know any examples offhand, but I wonder if anyone is using an inactivated coronavirus that would cause like a regular cold for this, like we do with adenoviruses. The problem is with these kind of common cold viruses, like some of them being coronaviruses themselves, using those as a vaccine is a problem, could potentially be a problem because we have already been exposed to these colds. And so we have antibodies presumably against them. And so it's not clear to all immunologists yet, and certainly not to me, what will happen when we already have antibodies generated and then we give a boost with this other vaccine, which could be a coronavirus itself that's been inactivated or a message or a subunit. So that's the purpose of these safety and efficacy trials, to see what happens if you use those other viruses like the adenovirus. But that's a good question. I don't know if anyone's using a less potent coronavirus itself as a vector. I'll have to look that up.
Some people who test positive are greatly affected and wind up in hospitals or die, and some that show no signs at all, or maybe just a day or two of symptoms. Do you see a difference in binding to the ACE2 receptor for these individuals?
I don't know if I really know all the answers to that. What people are suspecting is going on with children who do get infected and do transmit, but do not get ill, is that there's a different expression of ACE2, that there may be either less of it expressed in deeper parts of our lungs, or maybe there's less of it in general expressed. And then there could be variations among individuals of the type of ACE2 that's expressed, but I actually don't know of any studies offhand that have looked at binding efficiency of spike to different ACE2. I mean, for the most part, we all have the same kinds of proteins, and any defects or changes in the amino acid sequence usually means a disease of some kind. But it is possible. I don't know offhand.
Who can assist with an ingredient design of COVID SARS-2 protein subunit?
Who can assist with, say that again?
An ingredient design of COVID, sorry, my thing just moved. COVID SARS-2 protein subunit.
Sure, exactly what you mean by recipe. But we use a gene. So early in March, the entire sequence of the whole RNA that makes up this SARS-CoV-2 virus was published. And ever since then, many, many more sequences have been published of all the different isolates from different patients all over the world. And so there's one gene that's encoded in there that's for the spike. And so we use that gene that actually does become engineered over time. So we engineer it to help it stay together, to function well enough in our experiments and to best approximate what we think is on the surface. So lots of labs. It's actually pioneered by a lot of HIV labs who've done a lot of engineering on the GP120 protein of the HIV. So we use a lot of tools that we learned from other virologists to figure out how to change the recipe, really, I guess if you want to use that term, change the gene sequence.
Does each type of coronavirus have a different type of protein attached to it?
Yeah, the spikes are quite different to each other. Yes.
Which vaccine induces with mucosal immunity?
Oh, that's interesting. There are vaccines that are given as pills, or that as something that you would eat, like the oral polio vaccine, or there are vaccines that are given to the military. I don't remember what they're called, but they're actually in pill form, and you ingest basically these adenovirus vectors that protect the individual via mucosal immunity, meaning immunity that's generated in the gut, in the intestines. And so there are several examples, and those are just two that I can think of off the top of my head for mucosal immunity. And only recently on a This Week in Virology podcast, somebody was starting to talk about mucosal immunity and are we thinking about that? And the last I heard was, oh yeah, we really should think about that. So I don't know what the current update is, but that's an awesome question.
Great. Vaccines. Do you think COVID-19 infections are growing more or slowing down?
That's always been a complicated question because it depends on how much people are testing. So the positivity rate is a better measure of how many cases there are, new cases. And so on the CDC website, for example, you can track total cases, you can track case positivity or positivity rates as well. So meaning out of all the people who get tested, how many are positive? And so that's the number that would show you if outbreaks are occurring, really. So it depends. It's a little complicated now with tests becoming politicized, and it's hard to tell.
Here's another one that could be politicized. Do you think this vaccine will be mandatory for all people?
I don't know. I really don't know. Not every vaccine is mandatory, so just time will tell.
So here's a question about, I think, herd immunity. How many people need to get infected for people not to wear masks or even just not to worry as much?
So each virus has a different transmission rate, which is kind of encapsulated in this one term called R or R naught or R0. And you can use that R0 to calculate a percentage of the population that should be immunized. So for SARS-CoV-2, I think the most recent estimate is still 70% of the population that needs to have immunity. For something like measles, which has a much higher R0, it's something like 16 versus for SARS-CoV-2, it had been around 2 early on, and then we put in some measures to control it. So for measles, you need something like 95 or 98% herd immunity, and for something with an R naught of like 2, like for SARS-2, we think we need something like 70%.
All right, well, we are past time. We just had a couple questions left that I'll definitely share if you don't mind following up with those. But thank you so much for your great presentation and all the great questions and taking the time to answer these. We knew this would be a hot topic that everybody would want to know about.
Well, you're welcome, and thanks for your patience. There's so many things we still don't know.
Great. Clemencia, anything else to wrap up today?
Nope, you have till Friday to submit your papers, and that's it. So thank you, Emily.
You're welcome.
it out. Sorry to interrupt you, Emily, really quick. If you haven't filled out the survey for our speaker next week, please do that. He'd really appreciate it, and I think you'll be interested in the results. Thanks. Yes, thank you, Emily. Sorry for interrupting.
Yeah, you're welcome. All right, I'll answer these questions even after everybody leaves.
Great. Thank you.
Take care.
Bye.